So today is the 7th July 2020, and it's my pleasure to welcome Luca Musante. So he's an assistant professor. He's a part of UTA Erzgebirge team. So Luca started to study biochemistry of urinary markers in renal disease 24 years ago as postgrad researcher in Italy. His research interest there was focal segmental glomerulosclerosis, a type of glomerular kidney disease which leads to permanent kidney damage, and also the incidence of recurrence of this disease even after kidney transplant. So since then, he has been optimizing the use of urinary extracellular vesicles as a novel source of biomarkers of kidney disease. And today, he's going to share his expertise in urinary extracellular vesicle analysis and pre-analytical variability. So please welcome Luca.
All right. Hello, everyone. Thank you for the introduction. I would like to thank you, the organizer, for giving us the opportunity to present some published and unpublished data. So today I'm going to talk about urinary extracellular vesicle and to highlight some of the factors that our opinion can be of interest and important when we want to carry out a comparative analysis between different samples. This is the outline of today's presentation. After a short introduction, a brief overview of the method to isolate urinary Vs with particular emphasis on centrifugation, something about urine composition. Of course, we have to talk about Tamm protein or Uromodulin, the most abundant protein present in urine, and then the bulk of the study that was to address the question, what is the minimal volume of urine for downstream analysis and to be compliant with the MICEF of the myoflow cytometry EVs guideline. When we did this, we provide a full characterization of the EVs pelleted at 20,000 Gs, very often a neglected source of EVs. So as you might know, urine has always been since ancient time the ideal fluid to detect metabolic, kidney, and urogenital diseases. As a proximal fluid, it's particularly enriched of all those markers secreted by all the epithelial cells forming the nephron and the low urinary way. It's a relatively complex matrix with more than 3,400 protein, 1,400 endogenous peptides with a molecular weight cutoff lower than 10 kilodalton, more than 160 mature microRNA, and more than 445 metabolites. It's a very diluted solution and very dynamic. It quickly responds to the change of the environment. On top of this, we know that the kidney itself has its own circadian rhythm that work in synergy with the central system circadian clock, and both in healthy, but also during the disease. In fact, few years back was proposed that the proteinuria itself to follow a circadian clock. So in this slide, more or less, I wanted to sum up the most common method to enrich EVs. They can work alone, but most of the time, a combination of different methods can be used. So much so that the literature has been flourishing with dozens, if not hundreds, different protocols. There is no right or wrong protocol. Everything depends what is the goals of the study, what's the question that we want to answer. Of course, in top of the list, there is centrifugation in its different form, differential density gradient with different media, caution, centrifugation. And today I'm focused on centrifugation for two main reasons. First is the most used method, is considered and still was considered and still considered the golden standard for EVs isolation. And for me, I like it because it's relatively simple. What I mean, it's there is no physical barrier. So whatever you are looking for, if it's not in the pellet, is in the supernatant, or vice versa. In these slides, we want to sum up the principle of centrifugation. Of course, we can have a different type of rotors, different geometry, different volumes, and different size. We can, even if we don't have the right rotor, and we read in some paper that the study was carried out with a different one, there is the way to convert the parameter in such a way to get the same yield in the final pellet. This provided that all the details are written, or you provide all the details. This is why it's so important to be clear and transparent during the reporting. It could sound trivial and a bit naive, but in a comparative analysis, the first thing that I usually think is to run the sample the same volume in such a way that the length of the sedimentation path is the same for all the samples. The final yield of the pellet depends, of course, from the relative centrifugation force and counteract by the friction and the buoyancy. Buoyancy is the difference between the density of the particle and the density of the medium, while the friction is more related to the viscosity of the medium itself. So having said that, the first question that we have to ask ourselves is, what do we know about density and viscosity of urine? As you might have done, or use a dipstick, one of the many commercially available, we can measure the density. As a rough ballpark estimation, the color of urine itself could be already an index of, if it's a sample that is diluted, from the color. Very light yellow color is an index of very diluted sample, with respect to a more intense yellow that is give the idea of being a concentrated sample. Overall, the density is much, much lower of the density of our EV, but I like to think that working in a nanoscale, small differences, micro differences of samples that are at both ends of the range can have a big impact on the final result. And let me explain these slides a little bit, because what do we know about urinary pigments? Is the main pigment is urochrome, a small molecule of less than 600 Dalton. Is a catabolite product of bilirubin. So bilirubin and its glucuronate ester are very hydrophobic. This is the reason why we had some polar head. So overall is an amphipathic molecule, and we have an hydrophobic tail and an hydrophilic head, polar head here, and an hydrophobic tail. And this hydrophobic tail in an aqueous environment like urine is not very stable. So, and we really don't know how this hydrophobic tail can be associated. We can extend some rules that we know for surfactant or detergents, where if the concentration is above the CMC, critical micellar concentration, it can form micelles, or it can find stability in some hydrophobic pocket of proteins present in urine. And I think we cannot rule out that the phospholipidic layer surrounding our EVs can offer a hydrophobic environment where to find stability. In fact, what's the purpose of these experiments? So, as I said, is a small molecule, less than 600 Dalton, so it should be quite easy to get rid of it by simple dialysis by diffusion. So in this experiment, we wanted to know what was the molecular weight cutoff that can let the yellow pigment diffuse into solution. So we started from 3.5 kilodalton, and most of the pigments were retained. We move up with the molecular weight cutoff. The following step was 10 kilodalton, and still not too much loss of yellow pigment into solution. We could reach this dramatic effect when we use dialysis membrane whose molecular weight cutoff was 300 kilodalton. As you can see, the yellow pigment associated with something bigger than start to diffuse and sit on the bottom of the cylinder. As the dialysis keeps on, the bag is pushed up to floating, and at the end of the dialysis, all the pigments are out, and when we change now, whatever is left inside the bag has the same dialysis, as the same density of water. So yellow pigment associated with something confers some additional density to the sample. As far as the viscosity is concerned, to be honest with you, there isn't too much in literature. This is one of the first papers that I came across. It dates back 1918, and where they measured the viscosity of urine to test their instrument. From that point on, not that much. In more recent year, we in this group studied the viscosity of urine, and I found that one of the clinical important factor affecting viscosity was, not surprise, the level of proteinuria. So if you plan to carry out a study on nephrotic urine, of course, albumin can change the viscosity. Diabetic nephropathy, where we usually divide the sample in macro, micro, and normal albuminuric, in a comparative analysis, we should keep in mind that the level of albuminuria can affect the viscosity. Of course, this is during disease, in course of disease, but I'm concerned about proteinuria also in healthy urine, and more specifically in the most abundant protein, Tamm protein or Uromodulin. But before going into some detail, let me pay some personal tribute to the two scientists that discover this urinary mucoprotein. Interesting enough, Igor Tamm and Frank Lapin Horsfall were neither nephrologists nor urologists. They were virologists. And if we think in particularly, Dr. Tamm was looking for viral inhibitors able to inhibit the hemagglutination of mumps, influenza, and Newcastle's virus disease. And he found it in urine. And it's quite intriguing because we know the similarities between EVs and viruses. Already in this WebEVTalk, Dr. Whitwer and Loftal talk about the EVs and viruses, and I think next Thursday, Dr. Terry is going to speak more about viruses and HIV and EVs. So it's quite important and interesting. The protein was named nearly 10 years after the discovery. Here, this is the first title that you can find in PubMed, where they mention Tamm protein. 30 years later, with respect to the discovery, a new protein, uromodulin, a glycoprotein isolated from urine of pregnant women, has a very strong immunosuppressive effect. And only 2 years later, they found that THP, it was the same entity of uromodulin. So Here we are, Tamm protein or uromodulin. The most abundant protein present in urine is synthesized specifically from the epithelial cells forming the thick ascending limb of endless loop. It's synthesized as a GPI anchor protein. When it's translocated on the luminal side of the cells, proteases, hepsin, cleave the protein. As soon as released, the two ZP domain are able to interact and start to form dimer, trimer, oligomers, and filaments. Filaments that can reach some dozen of million Dalton of molecular weight and can reach the length of 20-25 micron, and they start to precipitate in a gelatinous cast already a very low speed. For example, in this was 4600 G, and you can see here the gelatinous cast of THP. In our EV prep, collected 20,000 G. You can see the polymer of Tamm and all the EVs entangled in its polymer. These are cryo-EM picture, and you can appreciate the heterogeneity of the EVs with roundish sausages-like EVs. We have EVs on the verge of be kicked out another EVs, and smaller and bigger one. So THP can be a confounding and a factor that can entrap EVs. So what are the factors that trigger the polymerization of THP? Temperature in vitro, but in vivo we have to consider pH, the concentration of sodium chloride, divalent cation, the concentration of THP itself, so the hydration level, and some unknown analytes, like, why not, the yellow pigment, as you can see in this yellow palette. So the following question is, when we want to compare different samples, is what is the ionic profile of urine? And in these studies that was carried out on the 24-hour collection, you can appreciate what's the interval range of volume from 1 up to nearly 3 liters. pH can span up to nearly 2 units of pH. Sodium chloride of our interest for the polymerization of Tamm. And it's interesting, would be interesting to know when we have to compare samples that, again, at both ends of the interval, how THP behave, how much is polymerized, and so forth. And this study, you have to think, that was made on the 24-hour collection. In the nephrology is considered the golden standard of urine collection. The interval can be even larger if we consider the spot random urine collection, as here representing, where you can see the breakdown of the 24-hour collection, where each void was kept separated from each other. And we are going to present more data about this in a virtual poster in the forthcoming ISEF meeting. So I don't spend too much time on this slide. I remind you to the ISEF meeting. So how is translate of these differences when we apply a differential centrifugation protocol? So one volunteer collected its own first morning urine for 4 consecutive days, so one after the other. And here we start from raw urine, where we load the equivalent of 0.5 milliliter. And as you can see, the protein pattern is just the same from sample to sample. Also, the amount of Tamm that we get in each sample looks very alike. But as soon as we start to process the sample, is just like to open a can of worms. All the habits come out. And you can see this is the palette that we obtained 4600 G. And unfortunately, we lost the very first palette. An accident happened, but you already can see in different day how much Tammosphal we collect in the pellet, very abundant in day 3. And this keeps on when we increase the speed to collect EVs as a 20,000 G and 140 G. You can see how much different level of Tammosphal, amount of Tammosphal there is in each sample, and probably also the EVs that we collect in each sample, in P20 and P140. And finally, the supernatant, again, as you can appreciate, the protein pattern is the same. Now, Tammosphal is not anymore the most abundant protein, but albumin becomes the one. Western blot analysis of some of the markers that we were interested at that time, and our podocyte marker, very specialized epithelial cells forming the glomerular filtration barrier. You can see different recovery in different sample in different day. And I wish I could avail at that time some reference material to spike in the urine at the very beginning of the centrifugation protocol, like the material that Dr. Hendricks presented few weeks back, to just normalize for the final recovery of the reference material and not for the final volume. We investigate a little bit more the differences between the two pellets obtained at 20,000 G and 140 G. More or less, the protein patterns look very alike, but for the amount of Tammosphal that sometimes is not present in the 20,000 G pellet. Conversely, we can see a very strong signal from TSG101, one of the most common markers to detect. The particle size distribution measured in NTA shows an overlapping particle size distribution with a peculiar shoulder in the range of 300–600 nanometer, characteristic of urinary Vs. Of course, from the quantitative point of view, with the ultracentrifugation, we obtain more particle, but we also apply 7 times more the relative centrifugation force. So since 20,000 G provide a good amount of vesicle, a lot of TSG101, we wanted to investigate further this pellet. And in the meantime, we wanted to answer the question, how much urine do we need to process to get enough EVs to carry out some downstream analysis? So we collect first morning urine, centrifuge for 5,000 RPM, that correspond to 4,600 G. And the supernatant, we use it in a different volume, starting from 0.5 milliliter up to 13.5 milliliter, to mimic more or less the volume of urine present in biobanks stored in ultra freezers at -80. We centrifuge 50,000 max speed of our benchtop microcentrifuge using microcentrifuge tubes 1.5 milliliter capacity. The pellet was characterized with several of the most common techniques used to analyze EVs. Let's start from cryo-electron microscopy. Again, you can see quite heterogeneous population of EVs, small, larger, electronegative, electrodense. Of course, we see Tammosphal polymers that can entangle EVs, but we can see also polymers of Tammosphal that stick with the surface of the phospholipid layer of our EVs. We have multi-composite or multilamellar EVs, as here shown, but we can see here better in this, of the size of less than 500 nanometer. But if we zoom in, you can see even better that this multi-composite structure happen at the order of half micron with 1, 2, 3, 4, 5, 6 EVs inside a bigger one. But this happens also at very lower size. This more or less 100 nanometer size vesicle, you can count 1, 2, 3, 4 smaller EVs inside one to another. And again, these multi-shaped EVs gives the idea that these structures are not static, are metabolically active, and it seems on the verge of splitting into smaller EVs. We measure the particle size distribution and number with two platforms, TRPS, QNano from Rhizome, and NTA, Zeta View for particle metrics. And as you can see, the particle size distribution is the same independently from the starting volume that we are reaching V, from 0.5 up to 13.5, both for TRPS and NTA. The particle size distribution, and we use here NP300 nanopore membrane. We didn't adopt any filtration step. We want to compare, we want to know what was the real particle size distribution. And interesting enough, I think was just by chance, both techniques show a median of around 180, 190 nanometer. Of course, the more you have, the higher is the concentration. And when you normalize by the volume of urine, we got a technical CV, coefficient of variance, around 15%. And of course, as you can see, 0.5 milliliter is already enough to work out both particle size distribution and number with both platforms. Western blot analysis of different EVs marker show that this is my favorite antibodies and my favorite marker, TSG101. 0.5 milliliter is enough to detect the signal. But for other, like CD9, we need to process a larger volume of urine. 4.5, probably 9 milliliter of urine is enough. Two nephron marker, colectrin, something like 4.5 should be enough. Podocalexin is a little bit more sensitive. So depending what is the marker and the abundance in terms of copy on our EVs, different amount. And we didn't detect it, probably was much lower than the limit of detection, both calreticulin and calnexin, and here are the two positive control, kidney tissue from rat and a pellet of human saliva. So then we wanted to know more about this pellet, so we implement a proteomic analysis. So the first step was to remove THP, and we use a different reducing agent, TCP, instead of DTT. Is much stronger, or the reducing power is higher than DTT, and it's already effective at 10 millimolar. The pellet of the full set of volume was suspended in 100 microliter. Before centrifugation was diluted in 1200 microliter of buffer, and this was the key for a successful reaction. And then the centrifuge, again 30 minutes to split the pellet and the supernatant. The supernatant for Western blot and SDS page was precipitated with trichloroacetic acid supplemented with sodium deoxycholate, and then mass spectrometry was used to characterize the pellet, and NTA for the particle size distribution in both fractions. As I mentioned in the previous slide, the key was the dilution, and the dilution a very low ionic strength. As here I highlight in red in two independent sets of experiment, and independently from the pH of the reaction. As here you can see, in low ionic strength, the tamosphere is released in the supernatant, while when we use higher ionic strength, tamosphere is retrieved again in the pellet. And probably for the presence of salt, we in an unfolded tamosphere for the reducing agent, because I didn't tell you earlier, but THP has 48 cysteine, and all of them are engaged in 24 disulfide bond. So when we use a reducing agent, the molecule open up, and it can be released in the supernatant, provided that we work at very low ionic strength. time course and THP dose response shows that the reaction is quite quickly or already at time 0. That is not a real time 0, but there is still 30 minutes of centrifugation. So you add TCP and then you centrifuge 30 minutes is enough to release THP in the supernatant. And when we check the full set of volumes in 3 independent experiments, 3 independent prep, with different amount of atmosphere, you can see that all the THP is collected or is in the supernatant and nothing is left in the pellet if it's not traces. Again, Western blot analysis confirmed that the majority of the signal is recovered here in red in the pellet rather than in the supernatant. Even when we test our most sensitive marker, TSG101, as you can see, traces are left in the supernatant. When we check albumin, more was in the supernatant and only traces in the pellet, and is the same for THP. Electron microscopy, again, of the pellet after TCP treatment confirmed the presence of a heterogeneous population of vesicle, larger one, electrodense, smaller one, saucer shape, and here again, multi-composite with some vesicle, smaller one inside a bigger one, here and here. As a personal note, looking at the picture of the cryo-EM, I would say I have the feeling that removing atmosphere is just like to remove a sort of stabilizer. The EVs try to form more aggregate to be more clamped. So for some studies to have atmosphere is not that bad. Again, we measure in NTA the particle size distribution before and here and after TCP treatment, and here we reported again some of the volume. And as you can see, there is no changing of the particle size distribution across the volumes, but there is not changing of the particle size distribution even between the 3 different fractions. P20, P21 after P21, P21 after TCP treatment, the supernatant, as you can see, the particle size distribution is just the same. So we know from the Western blot analysis that we retrieve the majority of the V markers in the pellet. Some traces of TSG101 are left in the supernatant. Conversely, the majority of THP and albumin are in the supernatant. This suggests that soluble protein, and this is already reported and known in literature, like albumin, and in this case, THP, in its own different isoform or aggregation polymerization, can scatter light and mimic particles. So is also for the NTA point of view, THP can be a confounding element along with albumin. Mass spectrometry analysis, we provide our core facility with the delipidated samples, and this is a duplicate, and this is a gel that we ran as a qualitative control, just to know what we provide to the MS facility. We started from 9 milliliters of urine to obtain this pattern and to have at least 1.52 microgram equivalent of protein to digest with trypsin. They apply a standardized protocol. The guys found that the peptides mixture was quite complex, and they opt for 3-hour gradient. 2 unique peptides with 99% of confidence and a false discovery ratio of 1.3%. Overall, we found more than 1,200 protein, and 95% of that are found also in Vesiclepedia as a repository and in subset of urinary vis more specifically. Gene ontology an ontology classification for cellular component using David algorithm. On top of the list, we can find extracellular exosome with a good score. So if you ask me, do we have exosome in this palette? The answer is yes, for the mass spectrometry analysis and also for some Western blot detection of TSG 101 and CD9 independently from the presence of THP or uromodulin. So exosome on the top list, and I apologize, you can't read very well this table, but it's just to show that basically in our list of protein, we found nearly all the protein forming the ESCRT machinery. Just a few were missing with respect to published data. Of course, we found THP as a GPI anchor protein. And here I highlight in yellow all the unique peptides coming from the analysis. And what was interesting, that we found 2 peptides that match in the sequence between the cleavage site of hepsin and the GPI anchor site decided here. So there was good chance that we could have a GPI THP anchor to our membrane, and when we carried out detergent phase partitioning, we could find that THP was found in the detergent phase as a PODXL, one transmembrane domain used as a positive control. So yes, THP can be part of the repertoire of protein in the EVs secreted by the cells. We can have an alternative secretory pathway for THP. Of course, we don't know anything else. We don't know what could be the biological role of this structure. And it can raise some question in using THP as a soluble marker to check contamination. So it is a bit tricky. Dr. Bonta in Dr. Ma's lab carried out the transcriptomic analysis, and she picked 4 microRNA described in literature. And as you can see, we could detect our target already in a pellet obtained with 0.5 milliliter of urine, and we cannot rule out that even lower volume can be enough to detect some microRNA. As appendix of the transcriptomic study, we carried out also the analysis in the pellet before and after TCP treatment, so with and without Tamm protein. As you can see, for all the markers that we tested, there is no difference between before and after the treatment, before, with and without Tamm. So this suggests that if you want to implement transcriptomic analysis, there is no need to get rid of Tamm. It doesn't bother, it doesn't interfere in the extraction. Flow cytometry analysis, we use some marker using the same clone of antibody that we use for the Western blot, and we carried out a multiparametric analysis using IGF-BP7 TIP2. These are 2 peculiar markers, one of the few FDA-approved biomarker to diagnose acute kidney injury. Tubular marker, collectrin, and a podocyte marker. As you can see, the higher is the starting volume, the higher is the concentration. We think that 3 milliliter of urine is the right volume to have a good signal to noise ratio. Of course, depends on the abundance of the antigen itself. And for very abundant antigen, like we didn't study this, but for like tetraspanin, CD9, probably you can use less volume. When we normalize by the volume of urine as starting material, we found CV, a technical coefficient of variance between 20 and 30% for all the markers, including an Exim5, but PODXL. And this gave us a bit of headache. As you can see here, although we normalize, we keep on seeing an increase of the concentration of PODXL. And it took some time to understand the reason why. And most likely, one of the main reasons is one of the hallmark of urinary Vs, at least collected at 20,000 G. That is, in the unstained sample, is an innate autofluorescence with a peak of emission in channel 5, or in the red for channel 5 in camera 1, with the excitation laser of 488 and 561, and still in the red, in channel 11 for camera 2, where there is the excitation laser of in the violet, in the red. So what was interesting, and we are particularly lacking in UVA, the imaging flow course facility is well equipped, and we could check the spectrum of the autofluorescence with the spectral flow cytometry, Aurora, and here you can appreciate the broad range of emission of unstained urinary Vs with respect to plasma Vs with no autofluorescence, or plasma Vs stained with calcined in AM, and this is the spectrum characteristic of calcined. So I have to say that this autofluorescence complicate a lot our analysis, and what it's come out and very interesting, it was that a double staining of the autofluorescence, in this case we adopt channel 11 with PODXL at low scatter, medium scatter, and high scatter. And here, when we applied the mask function of the software, we could see a perfect match of the signal originated from PODXL and autofluorescence in this composite where it matched. We use also some Boolean function using the morphology and the intensity of the spot to check how many double staining spots we have. And also here, in those few rare events of swarming, but we have two particles in the same field, for both we could see a perfect match. Of course, if you ask me the origin of the autofluorescence, I don't have a straight answer. I don't have a straight response to give you. But let me go back to the beginning of this presentation when I showed these slides. And it was interesting to see that depending on the color, depending on the density, we can see more autofluorescence where we have more yellow, more pigments, and less autofluorescence when it's more diluted. Of course, the concentration can be important. Where it's more diluted, most likely we could have less CVs processing the same volume, but also the presence of yellow pigment, and we know that are amphipathic. They might be in the plasma membrane of our EVs. And what's interesting, as soon as the urochrome is filtrated through the glomerular filtration barrier, it matches with PODXL as a specific marker of the epithelial cells forming the glomerular filtration barrier. Finally, when we extend the analysis from different samples or from different donors, and in this study we enroll 4 female and 4 males. As you can see, we could get a different amount of tamoxifen in the 20,000 G pellet, depending what were the condition of the urine from each donor. This is in line what I presented so far. Protein pattern looks very similar for all the sample. Probably these are 2 bands that are present in the female samples, and here I can figure out that we still don't know what the impact of the menstrual cycle on the secretion of urinary Vs. There is no studies about that. And when we apply TCP, we retrieve EVs in the pellet, while all the THP was released in the supernatant. We check also our most sensitive marker, TSG101. Of course, all these three blots were acquired at the same time using the same parameters, and we could see only a minimal amount of TSG101 left in the supernatant. We measured in NTA the particle size concentration, and we found a CV, a coefficient of variance of 25–30%, slightly higher than the technical CV. So in conclusion, as I show you, urine is a very dynamic biofluid. The composition change from day to day, from subject to subject. And the composition of urine plays a key role in the final result, how many EVs, what's the yield of EVs that we get in a pellet. And it's really important to find either the way to standardize, normalize the samples before starting to process, or to have some reference material to spike in each sample at the very beginning and use it as a normalization factor. You will agree with me that P20 is a good source of vesicles. It provides any type of vesicle, microvesicle, membrane particles, exosomes, and even if you don't have an ultracentrifuge in your lab, I would say don't discourage. Most likely you can have a benchtop microcentrifuge and you can carry out an EV study. What's the minimal volume? Of course, depends on the abundance of the target and the sensitivity of the instrument and or the antibodies in use. And here from 0.5 milliliter, RT-PCR, NTA, TRPS, detection of TSG101, up to 20 milliliter for the detection of nephrin, a very specific marker, not too much abundant in urine. Again, EVs, urinary Vs, or EVs in general, from any biofluid, from any cell culture media, are always full of surprises. Autofluorescence was a bittersweet pills to swallow. The multi-composite, multilamellar structure are amazing, and the nomenclature still has to find a name for those structures. Can be Matryoshka EV, Russian dolls, why not? The relationship between THP and EVs is still to be fully understood, and for sure more work needs to be done, and there is still a lot to study. Finally, last but not least, I would like to thank Sabrina and Nuta to sharing some pain and some during the development of this project, Vinila and Valeria for the transcriptomic analysis, UVA core facility, Joanne Lanigan always offered a shoulder where to cry when it was time to analyze the EVs in flow cytometry with the autofluorescence and the pain in the neck to carry out the compensation. Kelly for the cryo-EM beautiful picture, Nick and his guys for the mass spectrometry analysis. Dr. Okusa, chief of the division, sponsor, and thanks to all of you for your kind attention.
Thank you so much, Luca. That was a great presentation and a lot of work summarized in your slides. There are a few questions. Maybe we can start with Phil. Phil, you have a question there, right?
So Luca, this is a wonderful presentation. You are such a You are such a sweet person. We have to think of a project to do.
Okay.
You have a really tough European background of biochemistry, I can see.
Yeah.
So the Tamm Horsfall protein binds light chains.
Correct.
And we found that some EVs bind light chains.
Correct.
So what was the light chain situation among the proteins, and were any experiments done to see if light chains were linking the THP and the vesicles?
It's an interesting experiment to do. It's best of my knowledge, I don't know if anyone has done it. I know has been published that in some myeloma, urinary Vs, there is a cast of light chain building up on the EVs. So I wouldn't be surprised that the light chain can work as a bridge between THP and EVs. Yes, it's not easy also because EVs carry so many different type of receptors that there might be mediators. Also THP has been found to bind cytokines, immunoglobulin. We know that we have Fc receptor, polymeric receptor on the surface of the EVs. So there are so many proteins that can mediate the binding that it's not a surprise, yes. And I think it's not too much investigated this, and I hope one day to purify enough Tamm Horsfall and to use as a capture in affinity chromatography and to see what type of EVs or virus can capture. That would be interesting, yeah.
It was very nice work.
Thank you.
Okay, I do have a couple of questions for you. So one is, I noticed when you showed NTA measurements, you basically said that in the PSD before and after TCP, you don't see any change in the size distribution. But I'm wondering if the NTA would give you enough range to see that shift.
Yes, of course, when you set up the parameters to carry out the measure, you have you can't go with the power of the laser too high or too low. And this is still reflect the heterogeneity of the EV population. If you want to emphasize the smaller one, probably the scatter of the bigger one start to be a problem. And so this is the big problem of to work with EVs in biofluid. It's just the heterogeneity. It doesn't allow you to pinpoint or some sites without having the other that can affect. For TRPS, you can go down selecting smaller one, but of course, again, the bigger ones start to clog the system. So it's always, you have to strike the balance.
Yeah, you have to compromise at some level. But I was wondering, I mean, that's what I was wondering, if you have used also some other method for measuring besides the nanoscale.
No, I would say the TRPS NTA as a bulk, and then again the cryo-TM are beautiful pictures, tells you that the heterogeneity is very big. Again, especially in NTA, we don't know when in the scatter of the light what happens when we have this multi-composite structure with EVs inside EVs. I really don't know, and I cannot predict if these EVs scatter more light than EVs that are, let's say, empty. That is still something that we have to investigate and difficult to predict. Yeah.
So another question, another curiosity that I have is, so you talked a lot about day-to-day variation, but
Yep.
I'm wondering if you did an experiment where you choose your best markers, maybe of glomerular function, and check them in the same patient in different days to see how they really change.
It's, well, as I show here, the same subject collected in 4 consecutive days. So we want to say in a very short term, what could be the differences. And of course, depending of your lifestyle, depending what you eat, what you drink, what you do, it can affect the composition. And that is what I wanted to stress, is it's very difficult to normalize the composition of urine as so broad range. If we compare plasma and urine, just only the pH, urine is 2 units of pH in plasma with 0.3 unit of pH, you are already sick if it's not that. So that is the broad range, and how this broad range affect the technical variability. Just a simple centrifugation, viscosity, density, how THP polymerize, how much we get in one pellet, and we deplete the system of viscosity to collect more EVs in the following centrifugation step. This is, I think, since it's very difficult to normalize urine since the very beginning, probably the key would be to spike in some material, some references, just like in qPCR, you spike in cell 39 at the very beginning, and then you normalize with your delta CT. We need something like that for, I don't specifically for urine, but I think it's, we can extend to all the biofluid.
Yeah, because the problem is very similar. Yeah, no, that's true. Okay, we have a question from Carolina.
Oh, hi. Yeah, thanks so much, Luca. It's very neat experiments and also very well executed as well. So I think my question is something that is much needed for analyzing urine EVs from bioresources. So have you actually compared the fresh and frozen urine and see whether that makes a difference in terms of the EV content?
Yes, in all this year working with EVs, I don't have strong experiment, but I have to say it's better to store EVs in urine as rather than in enriched fraction. But if you want to, I have the feeling, checking in Western blot some marker, that if you enrich EVs, it's better to store as a pellet rather than solubilize the EVs and store as a resolubilized solution. For these studies, we carried out on the P20, all the time that we carried out a prep, then we freeze the pellet as a pellet. And then we analyze few weeks later, and we couldn't find really differences in the detection of biomarker. Besides, when we use ultracentrifugation, so we have to resuspend the pellet and transfer it. I could see that during time you have a slightly degradation of your antigen. You little by little, it seems you lose antigen. Yes. So if I have to choose and to store EVs as a pellet.
Okay. And how about the other factors like fasting and that sort of stuff? Have you, what's your thoughts about it?
Yeah, I provoke many times my colleague in the division and say we should have standardized condition, just like a metabolic cage. With all the participants drink the same volume of water, they have the same food, they have the same lifestyle. And I think there is some studies about astronauts that collect urine in their program and they publish, but was more in general urine proteomics. So yeah, that could be another key, is just to intervene on the environment to standardize factor to have less variability. Yes.
Okay, thank you.
I think we don't have other questions because Kalyani said that her question was also answered. So I think we can close it. Carolina, do you want to tell us what's next? I don't remember.
Yeah, so I think I will recap first today's talk. So we'll discuss so much about the variability between inter-individual and then day-to-day variability of urines. And then also Luca has shown so much about how to optimize the methodology, and then which methodologies it's okay, but we need to consider the downstream analysis, and also how we need so much of a reference material so that we can assess the variability of EV from urine. So